Bismillahir Rahmanir Rahim. Welcome to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University. Our video today will be about neurotrophins in atopic dermatitis. Allergic diseases in general come with annoying symptoms like itching, coughing, sneezing, and watery eyes, leading to significant physical and psychological effects. The precise underlying mechanisms of these conditions remain partially understood. There is convincing evidence indicating that neurons and other neurosensory structures are not only affected by inflammation but also play an active role in regulating immune responses through the release of soluble mediators, primarily represented by a family of protein growth factors known as neurotrophins. These factors, or neurotrophins, initially isolated from the central nervous system, were recognized as vital for neuronal survival, differentiation during fetal and postnatal development, and also ongoing neuronal maintenance in later life. Actually, neurotrophins are vital for bidirectional signaling between immune cells and neurosensory structures in the airways and skin. Disruptions in the neurosensory network are linked to pruritus in atopic dermatitis and also airway hyperresponsiveness in asthma, which are major characteristics of these conditions. Recent research has uncovered evidence of interactions between immune cells and neurons, highlighting neuroimmune interactions in which neurotrophins play a significant role. The neurotrophin family comprises four known members. The first one is brain-derived neurotrophic factors. The second one is nerve growth factor, and then we have neurotrophin 3 and neurotrophin 4-5. Neurotrophins interact with tropomyosin-related kinase receptors. Tropomyosin-related kinase A receptor has a high affinity for nerve growth factor. Tropomyosin-related kinase B for brain-derived neurotrophic factor and neurotrophic factor 4 and 5, while tropomyosin-related kinase C is specific for neurotrophic 3. And there's another one which is pan-neurotrophin receptor, which is called P75 neurotrophic receptor. Neurotrophins can be released from various immune cells, including T cells, B cells, mast cells, eosinophils, and macrophages. In this diagram, we can see that different neurotrophins can be released from different immunocells in itch and inflammation. So, like nerve growth factors can be secreted from mast cells, eosinophils, monocytes, T cells, and macrophages, while brain-derived neurotrophic factors can be released from T cells, B cells, monocytes, mast cells, eosinophils, macrophages, and platelets, as well as keratinocytes. Also, we have neurotrophic 3 can be secreted from mast cells, eosinophils, monocytes, and T cells, and also keratinocytes. And we have neurotrophic factor 4 and 5 can be secreted from mast cells, macrophages, as well as keratinocytes. Neurotrophins share a common structure and have overlapping functions. Each includes a signal sequence and a prodomain connected to the neurotrophin sequence. This indicates that a post-transcriptional process is needed to produce the mature protein and define their biological actions. The first identified receptor was the pan-neurotrophic receptor, or P75 neurotrophic receptor, which was initially recognized as a receptor for nerve growth factor, but later discovered to bind all neurotrophins. This receptor is primarily involved in regulating survival, apoptosis, neuron outgrowth, and migration following activation. In contrast, tropomyosin-related kinase receptors mediate unique biological actions through neurotrophin binding, involving dimerization and autophosphorylation. Tropomyosin receptors and P75 neurotrophic receptor can be activated individually or simultaneously. As regard brain-derived neurotrophic factor, in atopic dermatitis, immune cells that express brain-derived neurotropic factors include eosinophils, T cells, B cells, macrophages, monocytes, mast cells, and platelets. And out of all these immune cells, the effects of brain-derived neurotrophic factor release is best characterized in eosinophils. In atopic dermatitis, brain-derived neurotrophic factor is elevated in plasma, serum, neurons, and eosinophils. Brain-derived neurotrophic factor is released by eosinophils in close proximity to peripheral nerves and causes outgrowth and branching of nerve fibers. Also, brain-derived neurotrophic factor inhibits apoptosis and induces chemotactic migration of eosinophils. The second neurotrophin, which is nerve growth factor, is expressed in eosinophils, T cells, macrophages, monocytes, mast cells in skin inflammation. Nerve growth factor promotes inflammation by initiating a cascade of mediator release from eosinophils and basophils, which subsequently facilitates the recruitment of other immune cells. Nerve growth factor inhibits apoptosis and increases the expression of interleukin-4 in eosinophils. Also, it induces secretion of interleukin-13 in basophils and modulates basophil functions by enhancing histamine and leukotriene C4 release. Nerve growth factor also upregulates substance P and calcitonin-generated peptides, which promote neuronal sensitization. Also, nerve growth factor induces mast cell degranulation and promotes recruitment of eosinophils to nerves. Nerve growth factor also affects the recruitment of other immune cells to nerves in inflamed tissue and mediate neuroimmune interactions. Nerve growth factor causes outgrowth and branching of nerve fibers and is elevated in serum and eosinophils in atopic dermatitis and also correlated with disease severity. In untreated stressed mice with atopic dermatitis like allergic dermatitis, increased mast cell degranulation was found, which is a sign for enhanced neurogenic inflammation. The mentioned study also provided evidence of connection between stress and nerve growth factor expression with heightened levels of epithelial nerve growth factor, tropomyosin-related kinase A, and P75 neurotrophic receptors in stressed mice compared to the non-stressed mice with atopic allergic dermatitis. Neurotrophin 3 and neurotrophin 4 and 5 are less characterized than brain-derived neurotrophin and nerve growth factor. Neurotrophin 3 shares about 50% homology with nerve growth factor, brain-derived neurotrophic factor, and neurotrophin 4-5, and has its unique receptor, tropomyosin-related kinase C. It is released by keratinocytes, melanocytes, mast cells, eosinophils, monocytes, and T cells. And except for mast cells, all these cells serve as both sources and responders to neurotrophin 3. The interaction between neurotrophin 3 and nerve growth factor leads to mutual secretion in keratinocytes. Neurotrophin 3 is expressed in all CD4 positive T cells, while its receptors, tropomyosin-related kinase C, is present only in T helper 2 cells. T helper 2 cells involved in allergic inflammation like atopic dermatitis respond to neurotrophin 3 with increased interleukin 4 release, consequently affecting other atopic dermatitis-related cytokines. In atopic dermatitis patients, eosinophils demonstrate upregulation of both tropomyosin kinase C and the low affinity receptor B75 neurotrophin receptors compared to healthy controls. Neurotrophin 3 stimulation has been found to significantly inhibit apoptosis in eosinophils from atopic dermatitis and allergic rhinitis patients. Also, neurotrophin 3 and neurotrophin 4-5 induces chemotaxis in the peripheral blood eosinophils from atopic dermatitis patients. And upon neurotrophin 3 stimulation, eosinophils show significant increase in eosinophil peroxidase level. Also, in atopic dermatitis patients, mast cells have higher levels of neurotrophin 3 compared to healthy subjects, suggesting an important role of neurotrophin 3 in atopic dermatitis. As regards the neuronal effectors of these neutrophins, neurotrophin signaling varies among specific neuronal populations. For example, touch-sensitive myelinated fibers and Merkel cells rely on neurotrophin 3, while unmyelinated nociceptive neurons depend on brain-derived neurotrophins and nerve growth factor. Sympathetic efferent neurons are critically dependent on nerve growth factor. Also, neurotrophins continuously modulate the excitability of adult neurons beyond their developmental role. In human skin, nerve growth factor sensitizes nociceptors for several weeks, resulting in prolonged low-level ongoing pain when combined with local inflammation. Moreover, nerve growth factor increases both cowhage-induced itch and pruritogen-induced pain. Also, nerve growth factor upregulates brain-derived neurotrophic factor expression in primary afferent neurons, leading to increased sensitization upon release in the spinal cord. Neuroimmune interactions can thus facilitate essential allergic symptoms related to neuronal activation such as itch and pain. In summary, higher expression levels of all neurotrophins have been observed in atopic dermatitis, allergic rhinitis, allergic asthma, or acute rhinoconjunctivitis compared to healthy controls. This increased expression occurs either locally in inflamed tissue and immune cells or systemically. Brain-derived neurotrophins and nerve growth factor in particular are well characterized and associated with pruritic skin inflammation. Neuroimmune interactions in atopic dermatitis lead to increased nerve outgrowth and branching in affected skin, along with cytokine release from immune cells like eosinophils, basophils, and T helper 2 cells. This highlights the potential of brain-derived neurotrophins and nerve growth factor as mediators between neurons and eosinophils in atopic dermatitis, offering promising insights into understanding pruritus. And here is my reference and my source of information in this video, which was published in International Journal of Molecular Science, 2023 April, and its title is Neurotrophins, Neuroimmune Interactions in Human Atopic Diseases. Lastly, I hope that you have enjoyed this video about neurotrophins, and I'm reminding you again to subscribe to the channel and activating the alarming bell if you are interested in getting any new updates.